Morning, everybody.
Morning, sir.
Good morning, sir.
Good morning.
Morning, sir.
Do you see what I'm sharing?
Good morning, sir.
Good morning.
Yes, sir, we can see it.
Okay. So let's start right away. So please, good morning, good morning, good morning, good morning. So please mute yourself. Mute yourself, please. All right. For those on YouTube, now you equally see the slides. So last lecture we discussed cell cycle leading to cell division, and we have two types of cell division. We have the mitosis and we have the meiosis. I already told you mitosis occurs in a cell cycle because it's a continuous process. Once one cycle ends, it enters another cycle and continue like that on and on. Then we gave the stages through which mitosis occur. I equally told you interphase is not a part of mitosis, not a stage in mitosis. So it's a separate stage on its own.
No,
It's a stage where
no.
and
But there's no lecture there. But that one I've never done online class because before. Let me use this phone. Because this phone that has Netflix.
Ayeni, what is wrong with you? I said you should mute yourself. Ayeni.
I'm sorry.
Mute yourself now. You are sorry, sir, and you are still talking. Can't you mute yourself? So, we gave the two stages, the stages of mitosis, and we ended mitosis with significance or importance of mitosis. We now move on to meiosis, which is a reduction division process. And it's a process that takes place during gamete formation. So, and it occurs for a complete meiosis. We have two stages, meiosis 1 and meiosis 2. And we have subdivision of each of the stages into 4 as well. For meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. I equally told you meiosis 1 is very, very important because this is where reduction of gametes takes place, and there are other important processes during meiosis 1. For instance, in prophase 1, we have 5 substages. Okay, we have zygote, we have leptote, we have diplote, we have pachytene, and we have diakinesis. All these are very important. And also we have what is called crossover. And we have the crossover, we have the diaspora there. Let me sound this warning until you hear me. Anybody that is still talking while I'm talking, that is not muted, I will send the person, I will remove the person from the class.
Sir, please, can you tell Princess Adesuyi to mute her mic? The class is echoing.
If a dollar, what is the problem?
Princess Adesuyi, please tell us to mute her mic. The class is echoing.
Princess who?
Princess Adesuyi, Princess Adesuyi.
Yeah, what did he do?
Her
What
mic
did
is
she do?
on. Her mic is on. It is making the class to echo.
To do what? Okay. I think she can hear you as well. That's what I'm saying. Princess Adesuyi, your mic is on. You are not muted. I said mute yourself and you are causing the echo.
Mode of carbon monoxide here, but we have 2 moles of carbon monoxide there. So again, I will now divide this equation.
Where did you see carbon monoxide in this class?
So once you divide it by 2, that will be CO2.
I don't want to see your video. Okay, mute yourself and remove the video. I don't want to see your faces. Smart Odunaru. Remove your. Who is this Zoom user? Don't you have a name? Zoom user, if you don't have a name, I will remove you now. And change your video to your. I said, Zoom user, I don't want to see your video. Don't you have a name? Change your name to Zoom. I mean, from Zoom user to your real name, or else I will remove you. So today we are going on, we are continue the class with sex determination in man. How is sex determined in man? What determines sex in man? In many organisms, sex is determined genetically. That is, sex is determined by chromosomes, by sex chromosomes in many organisms, including human. However, sex is accompanied by chromosomal differences between sexes in terms of numeric, in terms of number, and in terms of structure. That is, we have chromosomal differences between sexes in 2 different ways. It could be numeric and could as well be structural. There are 2 kinds of chromosomes in all organisms. There are autosomes and there are sex chromosomes. The autosomes are common to both sexes. Both male and female have these autosomes in common. The sex chromosomes differ from male to female. In human being, there are 23 pairs of chromosomes. And these 23 pairs are made of 22 autosomes and 1 sex chromosome. 22 are autosomes and only 1 pair constituting the sex chromosomes. In mammals and Drosophila, females have 2 X chromosomes. That's XX. I will not attend to anybody raising up hands now until after the class and I ask you to ask your question. So Jamiu Tawakali too. When you're sorry, drop your hands. I will not recognize you until after the class. Whatever you want to say, write it down. Males have 1 X chromosome and 1 Y chromosome. So when you see a sex being denoted by 2 X, that is XX, that's a female. And when you see a gender being denoted by XY, that is a female. However, Y chromosome contains very few genes, but X chromosome contains many genes. On Y chromosomes, we have the testis determining factor. That is the gene that determines the maleness. They are on Y chromosomes. This TDF, that is testis determining factor, promotes development of male phenotypes. So it promotes the development of male phenotypes. So any male that you see and look like a male, that means the TDF on the Y chromosome, okay, is well developed. This is the karyotype of a human. Karyotype simply means arrangement of chromosomes, okay, showing their numbers and their structure. Arrangement of chromosomes showing their numbers and their structure. As we have in this slide, we have 23 pairs of chromosomes that I said occur in a
Excuse
human.
me, sir. Your slide is not moving.
Owade, I can hear your voice. Stop talking. Mute yourself.
Your slide is not moving.
It's what?
Your slide is not moving.
What happened to the slide? Talk now.
It's still showing sex determination in man.
That is appearing, sir.
They are not seeing it again.
It's just showing sex determination in man.
It's not moving. Is it moving now?
No, sir.
Yeah?
Not moving, sir.
No, sir.
On YouTube or on Zoom?
On Zoom, sir.
On Zoom.
You can see this slide now.
It's not the topic. Test determination. That is what we are seeing here from the slide, sir.
Oh, okay. Don't worry. I will let me drop it.
Oh, no.
Can you see it now? Can you see it now?
No, sir, we can't see it. It's still the same thing.
Sex determination in man, right?
Yes, sir.
Is it moving
Yes,
now?
sir.
It's not moving.
Yes,
Yes,
it's moving. No.
It's moving.
yes,
No,
it's
this
moving.
is. Can somebody read what is on this slide?
Yes, sex
Sex
determination in many organisms. Sex is
determined
determined
genetically.
genetically.
Sex is accompanied by chromosome.
Chromosome
If it
differences.
is not moving in your own side, it's your own network. Oh. It's moving.
It's now that it's moving, sir. It's not moving before.
It's okay. It's okay. Can mute your mics.
It's moving now, sir.
Okay, this is what I have said earlier. This is what I have said earlier. Now, this is where we have.
Please, I can't hear you very well, sir.
Okay. Karyotype of Iman. Karyotype of Iman indicating autosomes and male sex chromosomes. Now, I said karyotype is defined as the Hello, can you hear me now?
Yes,
can you hear
it's clearer now.
Yes, sir.
Yes, sir.
Yes, sir.
No.
Yes,
Clearer
yes,
now.
sir.
For those of you that cannot hear me, it's from your own end. Others can hear me very well. Hello, can you hear me now?
Yes, sir.
I can hear, sir. I can hear now.
Okay,
Yes,
let's
sir.
Yes, sir.
Yes, sir.
No.
mute yourself. Let everybody mute in or herself. Mute yourself so that we can enjoy the class. Mute yourself now. For those of you that cannot hear me, it's from your own end. Others can hear me very well. Hello, can you hear me now?
Yes, sir.
I can hear, sir. I can hear now.
Yes, sir. We can hear you. Yes, sir.
Mute yourself. Let everybody mute in or herself. Mute yourself so that we can enjoy the class. Mute yourself now. So karyotype of human indicating autosomes and male sex chromosomes. I said karyotype has to do with I said karyotype has to do with displacement of chromosome of human indicating autosomes and male sex chromosomes. I said karyotype has to do with I said karyotype has to do with. So when karyotype is carried out, it shows the chromosomes. The chromosome, the total chromosome of an individual, showing the number as well as the structure of the different chromosomes in the individual. When you look at this slide, so when you will see that it shows the chromosome are displayed from number 1 to number 23, and you see in each number there is a pair. There is a pair. So for instance, chromosome 1, there are 2 chromosomes there. Chromosome 2, chromosome 3, 4, 5, 6, 7, until the last one. So the 22 that you see occurring in pair, these are autosomes. And when you look at the 23rd home, that one, you have a bigger one and a smaller one. And these two are the sex chromosomes. These are the sex chromosomes. Though they don't have the same size, okay, unlike others, in autosome, you will see that for them to pair up, they must have the same size. You cannot see one bigger, one smaller in 1 to 22. But because the sex chromosome, one is X, one is Y, they are not homologous. But even when they are not homologous, they are responsible for determination of sex in human. So this individual is a male. This karyotype we are showing is a male because we see one X and Y. If it is a female, you will see the two XX, and those two will have the same size as well. Then in that case, they can pair up together. So you see that we didn't pair X and Y, but they are standing side by side. When they are, when the two chromosomes are X, you will see the two XX are standing together. So this is the karyotype of a male. Then, as I said earlier, only few genes occur on on Y chromosomes. Okay, and among those few genes are the TDF, that is the testis determining factor. A Y chromosome that has lost the TDF, that's, you will now see in an individual, the individual will be a male and may not develop testes. The individual may not have testes because the gene responsible for testes on that Y chromosome is missing. Okay. A Y chromosome that has lost. Now there are some other organisms that equally use chromosome to determine their sex. Okay, we have seen in human and in Drosophila, which is an insect, X and Y chromosomes are responsible for determination of sex in them. So in Drosophila, okay, a female is also XX and a male is also XY, just like for humans. These X and Y chromosomes are rather diverse across animals and plants, and the XY is present in only a small fraction of species. So it's not in all organisms that we have X and Y as chromosomes determining sex. For instance, in birds and Lepidoptera. Lepidoptera are the class of insects, the moths and butterfly. They are the Lepidoptera. We have Z and W as their own chromosome determining sex in them. So in birds and these Lepidoptera, males are homogametic. That is, the two chromosomes there are ZZ. For females, they are heterogametic. That is ZW. Okay. Now look at these changes now. Chromosomes in birds, for a male bird, the two chromosomes are ZZ. That is, they are identical. That's why they are said to be homogametic. Whereas females are heterogametic. That is, the two chromosomes are not the same. They are different. Now, if you now apply this homogametic and heterogametic to human and Drosophila, you find out that in that case, a male will be heterogametic and female will be homogametic because a male is XY. The two chromosomes are not the same. So that is hetero. And in human female, it is XX. So the female is said to be homogametic. So this is now reversing the case here. So this is what I've said. In humans and many other mammals, the sex chromosomes are X and Y, and females have two identical X chromosomes, making a female to be homogametic, whereas a male carries two different types of sex chromosomes, X and Y, making male to be heterogametic. So while a human male is heterogametic, okay, a male bird is homogametic. Again, a female human is homogametic. Then a female bird is heterogametic. Okay, a male bird is homogametic. In describing Y chromosome more, I told you Y chromosome can vary in size from one organism to the other as a result of pressure of repeated DNA sequences. Okay, actually, when you consider the Y chromosome with other chromosomes, it is the smallest. Chromosome and contains the genetic information necessary to develop human male characteristics. So as smallest as Y chromosome is, if it is totally missing, the individual may not develop male characteristics. And contain the genetic information. So these are heterogametic and homogametic in sex determination. So that's that for sex determination. Now we move on to let's move on to the other aspect of the course, which is that's that for determination. Now we move on to let's move on to. Can you see the new slide I'm sharing?
Yes, sir.
No, sir.
Can you see this?
Yes, sir.
Yes, sir.
Yes, sir.
So we have ended the heterogametic and homogametic of it. So now we know how sex is determined.
No,
So let us now move on to genetics.
Yes, sir.
Aspect of genetics, especially the reproductive aspects. Please mute yourself. So let us genetics. Aspect of genetics, especially reproductive aspects. Those of you that are not muted are causing unnecessary echo. Genetics is the science of heredity and variation. Genetics is the science of heredity and variation. And the principle of genetics originated in 1900 with the rediscovery of a scientific article by Gregor Johann Mendel Gregor Johann Mendel is referred to as the father of genetics. He lives between 1820 and 1884. Mind all these dates being given to you. Now, Gregor Mendel published his work in 1886. Okay, and he called genes factors, which are the carriers of hereditary information. That time, he didn't give the name genes. He only said there are factors that carries hereditary information from one person to another. The word genes was now used by somebody else in 1909. And this is Johansen. Johansen now called Mendel factors genes. And that is what has been used from then till now. The genes are the carriers of hereditary information. They are the carriers of hereditary information. So these are some of the people that work in the field of genetics. Okay, so genetics being defined as the study of heredity and variation of inherited characters. What is heredity? Heredity is the tendency of offspring to resemble their parents and to resemble each other. Tendency of offspring of the same parent to resemble each other and to resemble their parents. And variation is also the tendency of offspring to vary among themselves and from their parents. When you look at your siblings, you notice that there are some traits or characters that are common among you, and that either of your parents equally has it. And again, you look at yourself, there are other features which is only unique to you, but not found in your other siblings and in your parents. So this is variation. As there are tendency to resemble, as there are tendency for offspring to resemble their parents, there are also tendency to vary from their parents. So we have these individuals work in the field of genetics. Rakesh Rahan and his son, look at the two. You see the resemblance between the father and the son. And when you look at Padma Shiri again, look at the son and the father. They have similarities. Okay, there is resemblance between them. The resemblance between the father and the son. So this is the picture of Gregor Mendel. And Gregor Mendel worked on plants. Generally, he worked only on plants. And the plant is Pisum sativum. That's the garden pea. Pisum sativum. It's a legume. It's a garden pea. And this is what he worked on. So, our genetics is said to be relatively young, but there are other modern branches of genetics, such as cytogenetics, molecular genetics, phylogenetics, developmental genetics, behavioral genetics. So all these are new branches of genetics. Who was Gregor Mendel? Gregor Mendel was an Austrian monk who discovered the basic principles of heredity through experiments he carried out in his monastery garden. That is somebody that, you know what, those that are called monk,
Okay, they are the Reverend Father that you know, that seclude themselves, himself in a place where there will be no disturbance, no worldly, disturbance, and they dedicate their life to the worship of their Creator. They don't have wives, talk less of having children. Those are the male among them, and there are females too. They are called nun. They too, they are in monastery. So, in the monastery where Mendel was staying, he observed some features on the garden pit, which made him to be curious. Okay, he observed different traits, different characters on this Pisum sativum, and this made him to be too curious as to want to know more about the plants. However, when he was doing this, nobody reckoned with him. They even thought he was not mentally stable, but he continued taking, observing and taking record of his observation. So, and was able to publish his work. However, in January 6, 1884, Mendel died at the age of 61. So, this is the work that he published in 1866. And was able to publish his work. However, in January, the title of his publication is Experiments in Plant Hybridization, and he wrote it in the German language, but the work was later translated into English, and they found out that it laid the basis for genetics. So these are some other geneticists that rediscovered his work, because when he was doing it, nobody wanted to reckon with him. But the good thing is he published his work, and the publication was still kept somewhere. Even after his death, his publication is still available. So among those that rediscovered his work are Karl Korens, is a German, Hugo de Ries, okay, Erich von Schamak, which is an Austria as well. So this rediscovery of his work are now referred to as Mendelism or Mendelian laws of inheritance. Mendelian laws of inheritance. Okay, Erich von Schamak, Austria as well. So this rediscovery. Now let's see some key definitions being used in genetics. There are some words that are used in genetics. These works are peculiar to the field of genetics. So let's see some of them and their meaning while discussing or describing genetics. So we start with gene itself. The gene, if you remember, I said was coined by Johansen in 1909. That is long after Mendel himself has died. So he now defined gene as the hereditary determining factor. Gene is defined as the hereditary determining factor. Okay, it is also said to be the basic unit of heredity. Basic unit of heredity. That is, it is the gene
It is the gene that is responsible for the transmission of traits from parents to offspring. A gene consists of a continuous segment of DNA, and in eukaryotes, the gene occupies specific position on the chromosome. So, genes are basic units of heredity, and they are located on chromosome. So, where they are found on chromosome, okay, is called locus. So, plural is loci. When you are looking for a gene on chromosome, where it is found, wherever that gene is found on the chromosome, is called its locus. It's locus. So locus is a singular form, and loci is a plural form. So as you can see, this is a chromosome, okay, made up of DNA, and this DNA and genes are equally made of DNA. So by extension, we can say chromosome is also made of DNA because there are genes made of DNA on chromosomes. Another one is allele. Allele. Allele is also called allelomorph, and it is defined as alternative forms of a gene. Alternative form of a gene which occupy identical loci on homologous chromosome. Allele control the contrasting characters of same traits. So when for the expression of a gene, there are two chromosomes. For the expression of a gene, there are two alternative forms, and each alternate form is the allele. So there are two alleles in a gene, and each one is responsible for the expression of the gene. One of the allele is said to be dominant, and the other is said to be recessive. And the allele. So there are two alleles. Each one. So the two alleles in the gene, one is said to be dominant and one is said to be recessive. And the other is said to be. So these are chromosomes, the picture of chromosome, and you can see the location of a gene, which is the locus on the chromosome. So we say the alternate form of a gene. So we say dominant allele and recessive allele. So what is a dominant allele and what is a recessive allele? Dominant allele we always express. You know, when we say something is dominant, that means it is the one that will always be expressed. So the recessive allele will only be expressed in the absence of the dominant allele. If a dominant allele is present with a recessive allele, the dominant allele will be expressed and the recessive allele will not be expressed. Not being expressed does not mean it is not there. It is there, but it is suppressed. So dominant allele mask or suppress the expression of recessive allele. And again, dominant allele, to indicate that an allele is dominant, we use a capital letter, and to indicate a recessive allele, we use small letter. As in these traits, a trait is height. Okay? If a trait is height, that means you can be short or can be tall. You can be tall, you can be short in your height. So the characters are now tall and dwarf. So if you are tall for your height or dwarf for your height, and From experiment, it is seen that tallness is dominant, whereas dwarfiness is recessive. So you can now see the allele that we use in representing the dominant, capital T. Then for recessive, small letter t, as I've explained. Tallness is dominant, whereas dwarfiness is recessive. So you can now see the allele that we use in representing the dominant, capital. Another key term is genotype and phenotype. What is genotype? Genotype is the genetic makeup of an organism. So all the genes in an organism constitute its genotype. Then what about phenotype? Phenotype is the physical features, stroke appearance of an organism. Physical features, okay? Physical features and physiological features, because not all phenotypes are physical. They are equally physiological. Okay, appearance of an organism. So, what makes it different? Genotype is genetic makeup. Not all the genetic makeup of an individual will be expressed. Those that are expressed constitute the phenotype. Constitute the phenotype. So, phenotype is the expression of genotype. It is the expression of genotype. So, the genotype, there are some genes in the genetic makeup of an individual that may not be expressed. Okay? So, but they are still present. So, phenotype is produced not only by the genotype. So, expression of genotype is not only by phenotype, but also by the interaction between genotypes and environmental factors. So there are interactions between genes and environmental factors. So if somebody inherits tall allele for tallness, okay, 2 or 3 people may inherit allele for tallness, but their height will not be the same. That means the expression of that tallness will depend on the interaction between the tall allele and the environmental factors. We have seen two, identical twins that when they are said they are identical, that means they have their genotype the same, but appearance may differ because environment will interact with them differently. So, as it's been explained here as well, a trait is height. The phenotype, okay, could be tall and could be dwarf. So when it is tall, it could be the 2 alleles are capital TT, or 1 capital T and small t. And then we have dwarfness to be small tt. Small tt. There can be any other alternative because the 2 alleles are recessive. So, let me quickly, because this slide leads to the next slide, because we are coming in sequential something from dominant to recessive, then the genotype and phenotype. Then here we see phenotype being tall or dwarf, and genotype. In the genotype, we have 2 alternatives. For somebody to be tall, there are 2 alternatives. It's either capital TT or one capital T and one small t. But for somebody to be dwarf, there's no alternative. The two alleles there are recessive. So when we now have these two alternatives for tallness, how do we now indicate this capital TT or the capital T and small t? That is what takes us to homozygous and heterozygous. So a condition in which both the members of an allelic pair in the homologous chromosome are identical. They are identical. But this identical can either be dominant or recessive allele. When they are identical for tallness, that means that is capital TT. And these are dominant allele. So they are the same, they are identical. That's why they are called homozygous. Homo means same. And for dwarfiness, the two alleles too are equally identical, but that is small tt, small tt. But where one is capital T and the other is small t, okay, that means they are not identical. So that is what is referred to as heterozygous. It is a condition in which the member allelic pair in the homologous chromosome are not identical. So here we have one dominant and we have one recessive allele. One dominant and one recessive allele. And that is the tallness. The individual will still be tall, but the two alleles constituting that tallness are not identical. One is capital T, one is small t. So in such a situation, we refer to the individual as heterozygous. 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 Let's stop here for now. Abi, you don't have any other class?
Sir, we have CHM class. So we have
class again, Abi?
10 minutes ago.
Okay, we are stopping here. Do you have any question quickly? We meet again on Monday morning by God's grace. All right? I mean, you don't have any other class? Any question?
We have CHM class.
Any question? Any question? We are stopping here. Do you have any?
Get it?
Yes.
Like.
If you have questions, raise up your hand. Let's quickly hear that. Any question? Michael Akeredolu.
Yes, sir.
Okay.
So you said that identical twins, most of them, like, they don't actually have the same height because of some environmental factors.
Okay.
So how can the environmental factor like determine it?
Identical twins are
Yes, sir.
supposed to be the same in all ramifications.
Most of them, like, they don't actually have the same height because of some environmental factors.
Yes. That is interaction between the genes and environmental factors. Now, okay. Environmental factors are not just climatic factors. Environmental factors are not just climatic factors. Environmental factors also include the kind of diet and lifestyle the individual is living. Okay?
Yes, sir.
Diets can make them not to be having the same height. They may have different rate of metabolizing whatever food they take. And that one will equally affect the utilization of that food in them. So you can make them to be different. And again, even when we want to talk of climatic factors, suppose the two are identical twins, okay, and immediately they are born, one is taken to maybe UK, and the other one is raised here in Nigeria. So you can now see they are now raised under different climatic factors. In the UK, that one may just be eating junk that may, making him or her to be big and tall. And in Nigeria, you may not have access to most of those things. Right?
Yes, sir.
And even if you have, and in terms of boldness, you can compare the two. So these are some of the interaction that may make the two of them not to be, not to be similar, maybe in terms of height, in terms of their body size.
Yes, sir.
There's another. You can compare.
I have another question, sir.
Okay, see, Michael. Okay.
Okay. You said that for a gene, you have two alleles, and let us say tall and short. That for if the short is recessive, the recessive factor can only function when the dominant factor is not present. How can the dominant factor be absent?
Okay, see, Michael. Yes, the recessive can only be expressed in the absence of.
You have two alleles, and let us say tall.
So
If the short is recessive, the recessive factor can only function when the dominant factor is not present. How can the dominant factor be absent?
Remember, they are going to be contributed by the parents. If the parent contributed the small t, the father contributed the small t, and the mother contributed the small t, of course, the resulting embryo will be small tt. So it depends on whichever one that is being contributed. They are going to be okay.
Yes, sir.
Does that answer your question?
Yes, sir. Thank you, sir.
Now, I
wanted to ask sir.
You said you want to ask a question. You are raising up your hand.
Yes, sir.
Okay, I'm listening.
Yes, sir. Under the genetics you were teaching us, you were saying something about variation. So I wanted to ask that what are like the main mechanisms that contribute to that genetic variation in a population? Sir?
Come again. I can't hear.
Under genetics, sir.
Okay.
Under the genetics, you were saying something about variation under genetics. So I wanted to ask. So you were explaining about variation being you different. That there's some different traits that you might have that your family don't have. Sir?
Expression of
you said
under genetics. I understand. I can hear you. What I'm saying there is variation. Variation does not mean that character or that trait is just occurring in you. It may have been in your previous generation, but it's not expressed. But it's now being expressed in you. That's why at times you say, "Oh, this child resembles the great grandmother," or resemble the. So it shows that trait has been in the lineage, but may not be shown in your parents. It may have been responsible for by recessive allele. So, but now it's now been expressed in you. So that's what we are saying. We are not saying if it is not in the lineage and it's now showing in an individual, that can only happen if there is mutation, if there is a spontaneous mutation for individual to express it. That's what we are saying. But if it has been in the family, okay, it may not be expressed in your parent. But even when it was not expressed, they have the allele for it. Maybe the allele is suppressed, and they now contribute that allele in you, and you are now expressing it. So You may vary from your parents, but if you look at the previous lineage, they will say, oh, the child resembles, at times it happens like that. That we say, oh, the child resembles the great grandmother or the great grandfather. So that is the variation we are talking about. You are now expressing,
Yes.
right?
Yes, sir. Yes, sir. Sir, I have another question, sir.
What's the question?
Hello, sir.
Yes,
Sir,
I'm
you
listening.
said now you answered the first question. Now, under the second question I want to ask is that what are like the main mechanisms that contribute to that genetic variation, especially like in a population due to like the long-term relationship between them? Sir, I have another question, sir.
When we are talking about meiosis,
hello, sir,
and when we talked about the
sir,
the
said,
significance of meiosis,
now,
I said meiosis one, where there is exchange of chromatids between exchange of a portion between non-sister chromatids, that alone can equally lead to variation. So when there is a portion being cut from a chromatid and now joined with another homologous chromosome, it must be homologous. You cannot have crossing over between non-homologous chromosomes. Crossing over is between homologous chromosomes, but the homologous chromosomes have chromatids. So it's going to be between non-sister chromatids. So when there is reshuffling, that leads to right.
Get one scholarship grant where I want to apply for masters. But now 4.3.
So with this, I think
after you guys graduate like at least 4.3.
Crossing over between non-homologous chromosomes. So by God's will, we see again on Monday. The homologous chromosome. Thank you and enjoy the rest of your day. So it's going to be between non-sister chromatids.
Thank you, sir.
Thank you, sir.
That leads to right.
Get one scholarship grant where I want to apply for masters. But now 4.3.
So with this, I think
after you guys graduate like at least 4.3.
So by God's will, we see again on Monday. Thank you and enjoy the rest of your day.
Thank you, sir.
Thank you, sir.